Hello again. How you guys doing? Right, you guys alive and well and uh, hopefully invigorated to some degree. This is pre-lunch. I'm warning you, after lunch, you better be as invigorated because we're not done. I don't know, there's not really a natural break in my talk, so there's a place that we will break, and when we reconvene after lunch, I might go back a little bit if you guys have any questions or and cover maybe some of the subtleties that I had to cruise over. I've packed a lot in. That's totally not what I do, right? I mean, it's way out of character for me to not pack a lot in. But I did. I packed a lot in. And usually I say it's because I want you to get it all and I want to torture you. No, it's not torture, I promise. But I want you to get it all. And even if you didn't get it in this moment, it pressures you or it excites you or invigorates you to go and look deeper, even on your own time. That's true, still true, but the reason I packed a lot in today is because you will hear this again. What I'm going to tell you in a few minutes is why you'll hear it again and why you'll hear it in different overlapping moments. Let's use an analogy. So we know the microbiome is the conglomerate organ of microorganisms that resides within you, on you, throughout, and so on, right? It's that beautiful network of microorganisms that we can start considering its own organ, or it's as an organ in your body. Every bit as useful and necessary as any other. But we also know through the research that it's involved in a lot of different processes, right? And I mentioned a few yesterday. We know it's involved in female health and endocrine support and immune, and now this weekend, neurological, psychological, and so on. So it's this little amazing little organ that it has a lot of interfaces. Because of that, as we move forward in the microbiome world, and you guys are going to be part of the select few experiencing what we've been researching the last 10 years, really heavy since 2010, so really 8 years of concentrated research, a couple of others in more light reading, reading because it was a novel. The Human Microbiome Project didn't finish until 2010. So a lot of the information we have today that we take for granted in 2007 was sparse at best. There was very little research going on in terms of large university NIH funded. There was the Human Microbiome Project starting in 2007. But when you look back, when we first started introducing these concepts, that research, you couldn't find, there was no centers. There are now multiple centers in university wings associated with hospitals that are doing nothing but studying the microbiome. Where it started out as maybe 50 scientists, there are now probably over 1,000 studying the microbiome. The amount of money that's being spent on the microbiome, NIH funding in its heyday, and it might still be up there. I don't know. The funding has changed with the different politics and things. But not too long ago, let's say 2013, 14, 15, you were looking at $500 million easily researching the microbiome. So clearly, it went from not being known very well, and we went a little bit of the history yesterday, to it's involved in everything. Let's pay attention to it. And there's different motivations. What's our motivation for wanting to understand the microbiome? It's important. I want to know your motivation. Because it does affect everything, and it's a natural process. It's a natural relationship we have that gives us our optimal health and vitality. You strip it away, you're now no longer vital, anti-aging, living healthy, and living a very happy life, from tiny to elderly. It's a critical part of what we want to do here. And most importantly, it's a natural world that we are embracing, do we not? All the herbs we consume, the great foods, and the diversity, these are all things that now feed our microbiome as well as us. So it's part of what we already believe. It's part of the paradigm that we're a part of. It just becomes part of that narrative that we want to embrace. And we are embracing it. So you're on a journey with me today that is going to take and unfold over the next couple of years. And we'll get into that. So really quickly, you guys, let me turn this on actually. All right. So one of the things I want to go through initially, so that's the psychobiome. I mentioned yesterday, and I don't want to really get into too much of that, but remember, there's the neurological piece and then the psychological piece, right? The study of the mind versus the study of the actual physiology of the neuroendocrine central nervous system, immuno-neurosystem. So those are the neuro part, and there's the psycho part, which is the mind and the mood. Won't get into that too much, but our objectives today, throughout the next 2 hours, is looking at a lot of the research, and that's new research. I mean, remember, this is a very new field, and it's changing the way we think of things, of course. We want to touch on the gut-brain axis a little bit. We want to get into, the gut-brain axis is now the microbiota gut-brain axis, all right? It's the MGBA axis, not just the GB axis. And we can start adding to that, of course. We can bring in thyroid, we can bring in other endocrine, like the adrenals, right? That's part of this whole cycle. The hypothalamus, pituitary, adrenals, and so on, and we'll get there. So we're building off of this model, and every month that goes by, we add to this model. This is a very, very fast-paced science right now in this world. It's extremely fast-paced. There's a lot of people contributing, and it's really growing at an exponential rate. That's the other reason why I want to push the limits of what we're presenting today with you all concerning probiotic prebiotics and the next phase of our evolution in this world. And I'll get to that too. So we know that it acts on a number of systems. Today we're focused on the central endocrine, a lot of the neurological systems, of course. And then, of course, we have the name. And then there's that acronym that Nicole and Jack and Mojan have already talked about. And I'll touch on that a little bit at the end as well, because that's we want that to be the anchor for how you process through in your own clinic, to anchor you to the steps. And the reason for that is today I'm going to cover microbiome elements, but as you noticed in Dr. Mojan's talk, there's a lot of other facets. I want to remind you all, this is one brilliant tool that needs to be used with all of your other brilliant tools, right? It's best when you use it with things, you can use it alone, just like you can use other herbs alone when needed, but it really is going to be amazing when you use it with other tools. That being said, let's hit it hard. What I'm so excited about, and the reason I'm really pushing this, I've said 2 years, I'm going to try to even cut some time off of that. Because what we're bringing to the table over the next 18 to 24 months is unprecedented. It doesn't exist in the world. It's diversity. It's clinical human probiotics. It's not ferments. It's not random soil bacteria. It's not any of those things that have some health benefits, but they can't be directly linked to the health benefits that we care about, and the health benefits we can monitor, and the health benefits that we want to feed. So if you look at a prebiotic, many prebiotics feed probiotics, right? That's really their job, beginning with breast milk. And the rest of your life, that's why you want diverse fiber intake from various plants to feed the microbiota. Diversity is king under most circumstances. What we're going to talk about today and in the next few seminars is the diversity that we have been growing and cultivating and studying. So I'm going to not only present to you data that exists in the microbiome world, the human microbiome world from other labs and universities, but I'm also going to be presenting you today data that we have created in our own lab using organisms that nobody else has at the moment. And you will be the first to have these organisms and some of the prebiotics, believe it or not. That's the exciting part, you guys. This is going to change the way you think about probiotics. Just like yogurt and probiotics were around in the 80s. Do you guys remember that? Even in the 70s, there's some Japanese formulas. They didn't have the research behind them. They were thought to have various benefits, but there was really no real research. They just thought, well, there's milk, there's yogurt, these things, they've been passed down through generations, cheeses. There must be some health benefits, but the research was really sparse, and no one really was that excited about it until the Human Microbiome Project. Did probiotics become everywhere? How many probiotics do you think are commercially available to you now? Just a guess. Give me a couple guesses. Commercially, you can buy 24. 24 species of probiotics. I'm not talking products. Species. Sorry, that was unclear. How many probiotic species is what I should have said? There's about 24. And some of that 24, when you research them, aren't real. They've just, if I study a probiotic, let's say I pull out of your feces a lactobacillus, and I start studying it, and I find out that it's a lactobacillus casei. But I start studying a little bit longer, and I'm doing research on it, and I'm feeding it to people, and I'm getting back some clinical data showing that it's good for the brain, it's affecting anxiety in positive ways. If that species is studied long enough by me, I can put a designator at the end of it. Lactobacillus casei Shayne, or whatever. Although it is still Lactobacillus casei, and it may have some subtle differences, those kind of things are being used as different probiotics, and in some cases they are, in some cases they aren't. What I'm talking about are unique species, not Lactobacillus, not Bifidobacterium, and we'll get there. So what we're bringing to you, the science I'm going to go through, it's heady, but I want to set you up first with a little bit about the gut-brain. We bring in the microbiome, and then we're going to get into some research and really why you're here, because the tools you'll be able to take home at the end. All right, let's get into some of this stuff because it's amazing. So this is a list, it's not even a comprehensive list of what species we have over the next 18 to 24 months. There's over 70 here, species. How do they fit in? How do we want to roll these out into the world using or directing it through the research that many of these have experienced in the microbiome projects? So we've chosen, these are your probiotic names with your probiotic various species. All together, these products will contain over 70 different species. Now you'll notice there is another column here. These are the prebiotic fibers, oligosaccharides, polyphenolics, flavonoids, other unique feeds for these organisms. And the impetus for all this is the project, and a lot of people, when the Microbiome Project came out, we were already working on some of these, and we decided this is our opportunity to do something about this, to do something about, 1,000-year-old art of growing organisms that are human-based, not just dairy or soil-based and so on. These are true human organisms. This is the tools that you will have at the end of the next 18, 24 months. Unprecedented among these. So that's the most exciting piece of this, okay? That's extremely exciting. What does this offer us? Diversity, diversity, diversity. In the prebiotics, you've got well over 30 different types of plant fibers in different shapes and forms. Why do we choose different shapes and forms? I won't get into this a lot because I'm short on time, but when you look at how your GI is made up and how the organisms colonize the GI from the mouth all the way to the rectum, there's areas where they are more anaerobic, more aerobic, and they metabolize carbohydrates at different rates. So some of these are short-chain oligosaccharides. You've heard of polysaccharides. Some are really long, complex chains called fiber. They metabolize at differential rates because they feed bugs differentially. And in some of these formulas, you're feeding one bug so that he can produce things, or she, to feed the neighboring beneficial bug. They work together in these biofilms. They work together in these communities. And if you feed the first guy, the second guy gets the nutrients it needs, and so on. So there's a lot of research that went into this, and I'll get some of that later. So that's the really fun part. Well, prebiotas, these are just some of the characterizations of prebiotics. This is not an extensive list, but I just want to remind everybody that these polysaccharides or oligosaccharides come from a number of different sources. And I should mention right here that I've altered the slides subtly because every week I get a new piece of data from the lab or from the groups we belong to in the microbiome world. So I've altered these since I made the slides. These slides are available on your app right now. This set. The one in your book is slightly outdated. I'm sorry for that. I just had to keep these up to date. But in the app, you can go download all my slides. They're available. I think Tiffany did that for me earlier. Okay? So you can have these when you're done. So take good notes. Prebiotics. So what is the gut brain? We've kind of gone through this. I went some of this yesterday. You know, we first discovered this enteric nervous system in the 1700s. And simultaneously, we're discovering, you know, microorganisms. We didn't really have any relationships then, no correlations, but they were both being discovered about the same time by various scientists. And it proved to connect more. We started getting more, you know, theories around the second brain and mood. And that started to come about. We didn't know how, we didn't know why, but there were some relationships. In fact, ancient medicines talk about this a little bit throughout different medicines, so it's nice to see. We start unraveling the galt, you know, so anatomy and physiology is really taking off in the same era. And then finally, we start to encounter immunology. So if I jump from ancient science, not ancient, but, you know, a couple hundred years old, to modern science, when the microbiome project was beginning, there were fields in science and research and medicine that had hit a wall. Essentially, immunology. And I'm going to pick on them. We were now facing the highest epidemic of what kind of issues? Chronic. And what's the basis, or what do we think is the underlying cause of what I'm referring to right now? Inflammation, and what's responsible for inflammation? Your immune system, right? The immune system is central to that. We were running into all sorts of problems we couldn't understand. We couldn't get past it. They were not helping. The science wasn't helping us, was it? No matter what they did, they weren't helping us. They were treating symptoms. Drugs were being created regularly to treat symptoms, but we weren't dealing with underlying causes. Microbiome changed that. And then we were able to define the networks the microbiome is responsible for, and we'll get into a little bit of detail later. What are those? The gut-brain axis now comprises a complex network of enteric nervous system, sympathetic and parasympathetic divisions of the autonomic nervous system, and the neuroendocrine and neuroimmune components. That's key. Remember that paragraph or that sentence, please, because everything from here on out, as I refer to the microbiome and how it's controlling and healing and doing all these amazing things through and with the psychology and the neurology of your body, happens through these avenues. It's how it's happening. If the vagal nerve isn't the avenue, it's going to use immune cells to traffic information up to the brain. Or the brain's going to say, the vagal nerve is backed up or something's happened. I'm going to use cytokines going back through the neuroendocrine. I'm going to use hormone cytokines to go back and talk to the microbiome. These are all the different pathways your body has in communication with the bugs. So it's there's redundancy. There's an amazing network through these different pathways that are constantly in motion, communicating, sending so many signals. I can't even begin to talk to all about them today, but we'll touch on a few. So that is key to understanding the intimate relationship. And it's so intimate. It's so intimate. And we'll get into that. And then, of course, this is made up of all these organs and tissues you guys are, you know, infinitely aware of. I'm not going to go there. So we start exploring this relationship, and we realize that it innervates the entire abdominal. And when it does, and I have a graphic here in a second, the way that it's anatomically designed, the enteric nervous system, is specific to communicating with the microbiome. There's no other reason for it. And the immune system, the galt system, is brilliantly designed to manage the microbiome and or pathogens that can access you through that area. So when you start looking at it in more detail, you realize that not only is the communication happening in this brilliant interaction, but you're built your genetics are built to form a relationship with these bugs. So your blueprint, your DNA is already now built to create this relationship. The way this is built, it's now becoming more and more clear that this was part of the design. This wasn't an accident. This is design. Does that make sense? This is innate, intelligent design. All right, we connect through the vagal nerve, which we're now much more familiar with, and the ENS is over 200 million neurons. It's about the size of a dog's brain. So it's nothing to balk at. It's a nice size nervous system. It's quite large. All right, this is what it looks like. This happens to be, a section of the small intestine. Right here we have the rumen, so this is where the food and the nutrients are running through. And if you look at these layers, this is what I'm talking about. These layers are brilliantly designed to sense the microbiome, to pay attention to it, to allow your immune system to talk to it, to allow the central nervous system to talk to it, and to allow the enteric nervous system to continually be in constant communication with the microbiome. So it's very, very, very amazing that we have this design. And that's this cross-section of that design. Oh, I didn't warn Tyson, but I have a video for you. I was supposed to warn him. The video that you're going to see here right now, and I might have to describe, let me describe it real quick, Tyson, sorry. The video, what you're going to see is the first time we've ever captured, we being some other scientists, not me, ever captured an immune cell doing its job inside your body near the bloodstream. So this is an immune cell, a white blood cell, in real live animation. Not, or I shouldn't say animation, that implies not real. In real life time. They were able to capture it. Normally you look at cells, what? In 2 dimensions, right? On a slide or on a flat screen where they've just been fixed and you're looking at the organelles and the DNA. You're not seeing them moving. This is the first time it's ever been done, captured 3D in the real world system. Now you can play it. I'll try to point it out. It happens pretty quick. Look for a little white thing cruising around. The blue is sugar. It's feeding. There it is. That thing is, there's 2 of them in there, cruising around your body near the GI, feeding and looking and hunting for pathogens, making sure that everything's being taken care of, checking in. And this is another angle they're going to bring into it. You'll see this. You see it? It's cruising, right? Can you believe how fast things are going? Isn't that cool? There it goes again. So it's showing you all that there's blood flow here. So it's outside the bloodstream. It's in the tissue. Right? So it can go up here and go into the lumen and check out what's going on in the microbiome and so on and so forth. It's amazing. So when you think about that in your body and how that little guy can go out and measure and check out the microbiome and then jump back into the bloodstream or the lymphatic system, travel up to the brain and deliver messages saying, hey, guess what's going on down there? Right? It's almost like that cartoon that studies, that you see kids watching, the little animations of what the guys do in the stomach and talking to the brain with their walkie-talkies or whatever. That is an amazing realization that has now furthered the intricate relationship we have with those systems. Again, those systems that I mentioned in that paragraph. Let's keep this going. So, Human Microbiome Project changed everything. It changed our entire thinking of the immune system, and now has changed our entire thinking about bacteria in general, our central nervous system, our enteric nervous system. It's just really redefined everything. And the cool thing about it is we in this room, researchers, clinicians, people that embrace the natural medicine, the healing, the functional, whatever you want to call yourself, we have an opportunity with the microbiome. It is not dominated by the direct going after a drug, going after something that deals with a symptom. We have access to the microbiome. We have access to things that change the microbiome. We can modify the microbiome, which can modify your behavior. We can modify the microbiome, which improves your inflammation, and so on and so forth. Right? So this happens to be, the revelation is, this is perfect for us. This knowledge can change the way you really treat and deal with your patients, your family, yourself. And we'll get into the herbal component and the nutrient component later. So it's very cool. So it brought all these things into focus, and it really did bring, for me, it brought plants into focus. Because there's a lot of publications that come out in science. You guys know this, right? And sometimes they create conflict. Hey, vitamin C is good for you. Oh, vitamin C is bad for you. Hey, this worked to do this. Oh, no, it didn't, right? You guys follow this, right? I mean, science is dynamic, but it can also be controversial and it can be contradictive. Some of the explanation coming through all the herbalomic and all the work we've done with plants, we never took into consideration the microbiome. When you take a group of people and you run a clinical study, I don't care what it's on, food, nutrition, drugs, they're now having to debunk drug studies, like expensive drug studies. Why? Because the people's microbiome affected the way that drug was metabolized. And it does it in our world. So the microbiome is a consideration that we need to now consider for everything you read and everything you're doing, because it changes the dynamic of plants, nutrients, and so forth. All right, going forward, I wanted to give you guys a real quick visual of the density of the microbiome in the GI tract, which is where we're going to spend most of our time, and it's what we're worried about right now. This is just an elongated version of the ileum, cecum, and then down into the proximal colon and distal colon. These are cross slices of these tissues, showing you the blue is your actual tissue, the mammalian tissue. The red is bacteria. So you can see it varies too along colons, and it varies from person to person a little bit. But there's pockets that are higher, like the colon is 100 times more dense in microbiome than the small intestine. But it does change and it varies, and they live in various crypts and crevices, and they really change their environment. If you were to shrink down to a little person and go in there, you'd be in New York City one minute, and the next time you'd be in a rainforest, and the next time you'd be in a lake. I mean, it's very dynamic down there. Scary dynamic. It's really fun. And then these are just the illustrations. My point with this slide is to visualize the complexity, because it's pretty amazing. Here's the data we have now starting. This is our starting point. This is our benchmark or our framework to begin microbiota studies or our discussions. These are things you've probably heard over and over again from me or Dr. Pompa or other clinicians or speakers up here. We've talked about how the microbiome can affect histamine and how the microbiome can affect immunological or, we didn't talk much about female male, but we got epithelial barriers. That's a big one, right? Leaky gut, the zonulin and so forth. That's a huge one. We've always known that about the microbiome, how it can, if it's impacted, if it's dysbiotic, you get leaky gut, then you have leaky brain, and we've known that for a while. So these are where the starting point was for a lot of the research that we started. There's pathogen protection. The microbiome, a lot of these species, especially lactobacillus, they make their own antibiotics, right? They produce things that keep pathogens at bay and or can eradicate them way more efficiently than anything you can take. So a healthy microbiome, not only does it work for bacterial pathogens, it works for fungal pathogens, it also works for viral pathogens. So your microbiome is already well defined and developed to help you out way beyond what you can help yourself out because of the number of genetic components to the microbiome. Remember, 3 million genes? It has a lot of ability. What are some of the other fun things here? So we have, of course, it's there to extract nutrients, promoting different hormones. It can synthesize vitamins and so on. So there's a lot of things that we've known about the microbiome since the project, but that's evolved. Today you're here because we've taken this early science and we've driven down into how and why, and what are the differences, and why when we look at the microbiome of you all versus me and so on and so forth, what are the subtle changes, and how do we understand those changes relative to the health of the person? And that's where we're next. That's where we're headed. So what are the associated conditions when you look at the microbiome and look at the impact it has on health? What are the conditions when you don't have health and you have the opposite of health? You have leaky gut. You have inflammatory diseases, very chronic. You have toxin assaults. The microbiome processes a lot of toxin eradication. They can metabolize toxins and get rid of them. So when your microbiome's healthy, a lot of what you're exposed to in the environment, mycotoxins, biotoxins especially, your microbiome is that's the first vanguard that gets rid of them, and you don't even know you've been exposed because it's done its job. You don't have a good microbiome, your life is a different life now. We know it's intimately involved in cancer. Healthy microbiome has been shown to change the way cancer can develop and or progress in the body. And there's a number of cancers that are related. Dysbiosis, of course, creates a lot of issues with just the gastrointestinal, and that can lead to further issues down the road, especially untreated. Some of the other things, it protects you from pathogens. I mentioned that. If you didn't have a microbiome, you wouldn't have an immune system. How do we know that? What's one thing I told you guys yesterday about that demonstrates or allows us to test that hypothesis and demonstrate it? There's a type of animal. What would we call it? Germ-free. Germ-free animals have shown us that with that, and we shouldn't call them germs. That's just not nice, right? But bacteria-free animals or microbiome-free animals, they don't develop an immune system. Do you guys remember the boy in the bubble? That's a real person. You guys remember that? And that he was born, unfortunately, in an era where microbes were considered dangerous. And the only way to deal with bacteria was to what? Kill them. Kill them all, right? Well, unfortunately, that was his undoing. So when he was born with that condition, it's a genetic condition that essentially inhibits immune system dramatically. I mean, his white blood cells and everything are terribly developed. Looking back now, researchers are now saying in the microbiome, and these are recent discussions in reviews, that we probably could have done something about that now, knowing the microbiome. But we didn't. We put him in a bubble, and after living there for a number of years, he was getting depression, anxiety. I mean, he was just psychologically was a mess. Isn't that interesting? Right? He's got no microbiome, and he's psychologically a mess. And then you add to that solitary confinement. No love, no touch, no compassion. None of those things that, by the way, drives a healthy microbiome. Touch helps a microbiome. I told you about the bullying case. All that interrelationship, the more you have a good social relationship, the stronger your microbiome is. That is another very clear bit of research by a number of researchers. You have a healthier psychology, you have a healthier microbiome, and you have good relationships. And it's a vicious cycle. You get in the positive vicious cycle, you're going to be healthy. You get in the negative, you're going to be in trouble. And it's very dangerous. So getting back to that's important. So we want to do, and then of course aging. The microbiome is central to aging, and it's because it transforms everything that goes into you. If you're not transforming them properly, you're not getting good nutrients, and your microbiome is that window into your body. All right. So there's a systemic influence of microbiome over all of the health. Central to this powerful and elegant relationship is how it affects all aspects of our senses, including happiness. That's my quote. I don't know if I like it, but I wanted to kind of pause here and remind you all that it's important. I'm always up here espousing the science and espousing, all these great things about what I love, the bugs, the food, the way it affects your physiology, etc. But honestly, we are here because we want to be happy, healthy, and we want to have relationships with our significant people. That's why you're here. I mean, you can be like that boy in the bubble, which I didn't finish the story. He wasn't happy not because he was sterile. He wasn't happy because he couldn't have a relationship because no one could approach him. So they tried doing something. They tried giving him his sister's bone marrow to give him an immune system. It wasn't compatible. She had a virus. She had a normal, healthy microbiome virus, not a dangerous one. It killed him. And that was very sad. That virus shouldn't have killed him if they would have worked on his microbiome. Of course, that's 20/20 hindsight, but still, we have opportunities that's an extreme case, but right now we have these opportunities to deal with threats that you guys see on a daily basis. All right, cruising on. This is another important graphic, and I'm sorry it's a little bit small, but when you download those, you can zoom in and check it out. This is what does the MGB stand for? Microbiome gut-brain axis. I want to start highlighting some specific processes in the microbiome gut-brain axis. I want to start to really, I don't want to say get into the minutia, but I want you to start understanding for your own, well, for me wanting you to do it. Too bad. I'm up here, you're down there. I want you guys to try to understand that there's really amazing pathways that are happening here, and they're really cool. And again, it's brilliant design. So you remember I told you about the enteric, the immune, neurological immune system, the central nervous system, sympathetic, parasympathetic, as well as the, well, I said enteric, so the vagal nerve. I've probably forgotten one, neuroendocrine. All these systems are represented here. This is neuroendocrine. You have neuroendocrine cells in the epithelial tissue that when they register information from the microbiome, they can send messages. They send chemical type hormones, as shown here, up to the brain. They can travel through the vagal nerve. They can also travel through the central nervous system. Either one. They have a choice, depending on the type of information it's sending. We have the immune system, right? We have a dendritic cell. It can reach out, see what's going on in the GI. If it sees a pathogen, it can send that message. That message, it can either travel, it can itself travel. You saw how cool that was, cruising around. It can travel to the brain. When it does, when messages get to the brain, like this guy, the dendritic cell, how do you think they convey that message to the brain? What axis do you think is one of the axes that is central to this phenomenon? Yeah, the HPA axis. That is central to this phenomenon. So by the time the message gets to the brain, the HPA axis is one of the axes that's central to what your body's going to do about it, the decision making. Okay? The amygdala is also involved in some cases. But this guy can travel up there. What else can this guy do? I mentioned this guy yesterday in my intro. Dendritic cell. What else can it do? It can grab a microbiome. It can grab an organism and carry it somewhere. Where does a dendritic cell carry microbiological probiotics in a woman's body, or women? That's the one that carries it to the breast tissue and delivers bugs to the baby. It goes down into your gut, picks up some passengers, takes it up to milk ducts, drops it off, and then your baby gets it. How cool is that? Can also deliver these things to other parts of your body, telling your body, we've got a problem. I've gone down here, I reached out I opened up the tight junction, which it wants to do. That's a normal phenomenon under healthy conditions. Grabs a couple of pathogens down here and says, hey, this isn't right. We've got dysbiosis going on. I'm now going to carry these up to the brain and let the brain know, five bell alarm or whatever you want to call it. It's an alarm system for you too, to help you with your pathogens. So there's our immune. We have the vagal nerve that is innervated with your endothelial tissue, and then of course we have the central nervous system. So all these systems simultaneously working to keep your garden growing and to keep you healthy and keep everything flowing back and forth. And it goes both directions. So there's something called afferent and efferent. That's simply, this diagram is just simply illustrate that messages go from your gut to your brain and vice versa. How many of you know people that when some traumatic stress happens to them and shortly after that they have GI issues? We know that happens, right? That's the efferent. That's the message going from the brain out to the tissues, creating a problem. And it can be positive or negative. It's used, that's just the trafficking information. Very important that you understand that. And I want to also point out here, this guy is central, which means when these messages are carried by one of those systems from the microbiome to the brain, it goes deep into the brain and starts letting the hypothalamus know what's going on, the pituitary, and so on. So you get hormones being produced based on information coming from your microbiome. So what does that mean, you guys? What I want to discuss just briefly, because I'm not sure how I'm on time, but I want to discuss briefly the implications of this scenario. What is the HPA axis important for? I mean, just list a few. It's an amazing axis, right? Fight or flight. What else? Pardon? Addiction. Thank you. Depression. Hormonal control. Look at the hormones that these guys produce. Does the HPA axis control biorhythms in women and men? Absolutely. Does it control fertilization? Absolutely. Does it control metabolism? Absolutely. So now what I'm trying to really get across is that this microbiome has access to the central computer. And it can change the way that central computer behaves in a number of ways. It can be positive or negative. There are organisms, I don't know if I have them in later slides, but there's a few, they're called Clostridium. You guys heard of Clostridium before? What's the scary one? C. diff. There are other Clostridiums. There's one that I have that you guys are going to see in the future called Symposium. That's a good guy, right? It can outcompete the difficile, right? It's a good guy. It wants to push him out of the way and say, no, you don't belong here. I do. I'm made to live here in the GI, not you. What happens, though, is when you have a bug like that, you have let's say, a Clostridium or a Enterobacter ACA, which are also considered pathogens, they love certain foods. They drive your cravings. And we've always wondered how they do that. How in the world can the dysbiosis, which is high in Clostridium and Enterobacter ACA, make me want to eat French fries and donuts? Doesn't that seem ridiculous? Yeah, when I say that out loud, it sounds ridiculous, doesn't it? That microbiome down here in my colon as well as my small intestine are driving my desire to go eat terribly. Do you think that's a real thing, or am I making that up? It's a real thing. It's been now studied. Those 4 systems I talked to you about, they've monitored how certain species of Enterobacter ACA, when you start a lot of sugar, which they love, pure sugar, they don't deal well with the carbohydrates we want in our body, but they love sugar. They bloom, right? They have what they need, so they grow rampantly. And then they start triggering the hormone through the central and the vagal nervous system, sending messages up to your brain, limbic system even, saying, please go and eat this. Please get in your car and drive to 7-Eleven or whatever. And it works because when they sever, you can do this in a number of ways. When they sever the vagal nerve in animal models, the craving stops. When they give the animal antibiotics, the craving stops. When they reintroduce those pathogens, the craving comes again. Nothing else changed. So they've looked at it from different angles, and it's absolutely true. And you can do that in a number of ways. That's the issue with pathogens. So the pathogen model, which I'll get to in a minute, is still one of the things we need to focus on. You need to focus on as clinicians. Is this microbiome is all well and good, but there's still pathogens we have to be really, really concerned about because they can change that. And when I get to the pathogen model, remind me, I'll tell you a story. All right, so that's the afferent, efferent. Now that we have this history, I'm trying to build the story of, we start out with the gut-brain axis. We've known about that for a century. Now we start understanding the importance of the gut-brain, the second brain, et cetera. Now we introduce microbes. So now we have the microbial gut-brain axis. We're starting to understand what it does for us and the way it interplays and communicates and all the different cool systems that are designed for it. Let's talk about when things go wrong and how we can now associate, for the meeting today, issues with the microbiome and how that leads to neurological and psychotic issues. And this is a short list. They're highly associated. And you could argue chicken and egg on each and every one of these. You could argue that you get dysbiosis and you end up depressed. You get, you will find yourself being diagnosed with bipolar, and 6 months later, you have IBS. These all exist. These are all real cases because there is an intimate, unfortunate, but there is that relationship. That's where we link. We can no longer ignore or just theoretically draw this line between our gut and our brain because it's real and it's really impacting so many people now. So many people are being impacted by these things, which lead to these things. And I'm going to tell you the pathogen story now because I'll forget. And the story relates to this slide as well. But I have a pathogen slide. Just remember that when I tell you the story. I want to give you a story that happened in, I forgot, I apologize, I don't remember the name of the city, but in just recently, within the last few years, in Canada, there was a outbreak in a small town, 5,000 people. They had a water contamination. And it was jejunii, Campylobacter jejunii. and it of course it created a big stir, because when you have 5,000 people get sick at once, that's nothing to be ignored. They had to go in, so you had the equivalent of like the CDC, and you had this microbiome researchers really just converge onto this city and go, what is going on? What is happening here? Why do we have the hospitals flooded with people with diarrhea and vomiting and dehydration? So they had all those symptoms, right? And they found in the water system this contamination. And there was also potentially some Escherichia coli, but the Campylobacter jejuni was in everybody, all 5,000 people. Now, they were able to get it under control. How do you think they got under control for a number of people? Right, that's an acute infection. What did they do? Antibiotics. Now, some people refused antibiotics. Well, because it was such a big outbreak, microbiome researchers were fascinated. They thought, you couldn't get a bigger clinical study. Let's follow these people. Let's see what comes of this. So they did. And what they found is after about 6 months, all of the organism was gone from them. They no longer had the jejuni in their system. So whether they did antibiotics or not, they were pretty much back to normal, right? They had beaten it, and they no longer have the infection. However, at about the 6-month mark, what do you think starts happening? Let me point it out to you. So we start out with diarrhea. We have a little bit of, maybe diarrhea, a little bit of reflux, and what I don't have here is throwing up or purging in that way. But what started showing up now is IBS, IBD, Crohn's. These things started showing up in this population. There was nothing else that would have led to this, and it was happening. So you take 5,000 people, now 1,000 of them have IBS. That isn't something you can create. That isn't a statistical anomaly. That's real. That's because of the infection. And the people that got treated with antibiotics had it way worse, okay? They weren't responding to any treatment. And furthermore, after about a year, guess what happened? What was their next crisis that they faced? Mental illness. Exactly. Depression, schizophrenia. They haven't lasted long enough to see if this comes into play. This was certainly there. Anxiety, like crazy, stress, PTSD. These mental illnesses now came into play. And again, it was a giant part of that population. So the lines were clear. They were not, you couldn't ignore it as being, oh, one guy got it in 5,000. No. We're talking thousands of these guys, men and women got it, and kids and so on. So that is a huge, huge case study that you could never mimic, or you would never ethically ever want to mimic, actually. Getting 5,000 people sick, how horrible that'd be. But look what they've learned. These connections and the progression, by the way. We've talked about chronic diseases taking time, type 2 diabetes, and so on, how they evolve over time. And there's a lot of ways to start them. This was started by a pathogen. But keep in mind, when you're dealing with someone further down that spectrum and you're starting to deal with mental and or serious SIBO or IBD, IBS, Crohn's, remember that they're connected and they go back in time. You need to get them back to health. Right? It's taken some time to get there, and now you know the connections. You know this has got to be dealt with, so does this simultaneously. You can't, and then what's in between, all those 4 pathways I talked about in between, those are the 4 things you focus on. Immune system, enteric system, endocrine system, and central nervous system. Does that make sense? Because they are the bridge. They're the ones that are perpetuating this phenomenon. So we got to get them all better. So as Hippocrates said, that abdominal viscera were regarded as the principal set of disease in mania. So way back then, he related your gut to your mania. And in short order, what this functional guy was saying is, let's not ignore the fact that because we don't have an answer, that we call it, I think, what did he call it here? He called it metaphysical. I don't care what you call it. If we don't have an answer, don't ignore it, because it's real, and it will someday have a description. So this is what I would term as intuition or other metaphysical things that lead you to an answer that you're just waiting for science to catch up to. Does that make sense? All right. So a footnote. I wanted to throw this in there. It's something called functional psychiatry. I don't know. This was a footnote in a psychiatric publication because this microbiome dilemma related to neurological and psychological distress and disease and so on, it hasn't been embraced by this community at all. In fact, how many of you guys have heard about it from mainstream people? No one, right? And when you look at it, when you look at the possibility of reversing it, it doesn't fall into the bailiwick of what they normally go to, their go-to stuff, right? SSRIs and so on. That isn't the solution here. So this was a footnote in a journal, and I thought it ironic because at least somebody in the corner, now it wasn't the headline, unfortunately, but in the corner, they were at least admitting this is a possibility. This is the future. This could change things for these guys quite extensively, whether it be psychiatry, psychology, and so on. There's a connection that is no longer ignorable. Do you guys agree? Agree? All right. I know this is heavy stuff. So what they're learning in psychiatry is that both affective disorders like depression, anxiety, trauma, stressors, PTSD co-indicate with an increase of inflammation, which is a risk factor, and that inflammation is mediated by the gut and the immune system, as you know. It appears that microbiota is necessary for the normal development of the amygdala, HPA axis, and the frontal cortex. This is a note for me to remind you, because I'm going so fast, and again, you'll hear me later, and we'll continue down this path together. We'll keep going down this path of microbiota. That little paragraph, back to my germ-free mice, and now that's moved into humans. So these were preclinical. Since 2010, a number of clinical studies have come out. Right? Before that, it was all animals. But now that we've just, started to understand it in animals, it's moved into humans quite rapidly. And what they've shown is that if you do not have a healthy microbiota, you do not develop a healthy HPA-T axis, you do not develop a healthy amygdala, and you do not develop a healthy prefrontal cortex. That's critical, is it not? Without those three, you're doomed. It is very scary to think that our children, if they do not develop those three things with a healthy microbiota, we're going to be looking at children with a lot of issues that we don't understand and that we've never seen before. That's critical. And I wanted you guys to point that out because these studies on human and animals show that without that microbiota, if you strip that microbiota, you literally don't get the development. I'm talking the tissue, the neuronal connections, the plasticity. None of that develops appropriately. When you reintroduce into animals, so we can start to look at this, and it's starting to happen in humans, but when you reintroduce into animals, healthy organisms, some of which we will talk about at the very end of this, guess what happens? You do start to regain the development of those three brain tissues. So is that not an amazing silver lining? That you can reverse this. This isn't your destiny. We say that a lot, but it isn't your destiny. Very cool. I was excited. It's most effective in the younger, but it's still effective in the older, right? And cognitive goes in there as well. I don't touch a lot on that, but cognitive goes in there. And things like autism, which you're going to learn more from Dr. Pompa and Lisa and other speakers, by the way, about some case studies and how they're really teasing out some of the really cool things in the clinic. So although I won't give you some of that, you will get that the rest of this weekend. But one of the things that's really fun is as you see this dynamic and you can reintroduce these bugs, and even with few bugs, by the way, you take a small cocktail, 3 or 4, you get a benefit. That's amazing. That's the silver lining we have available to us. And it's available to you guys. I mean, this is really amazing. Okay, moving on. All right. So potential mechanisms have been suggested about how they influence depression. I've covered most of these. There is some IgA and IgM-mediated inflammatory responses. So now we're starting to understand what the bacterium are doing in your gut and how they're influencing things like inflammation and your immune system specifically. Generically, I tell you that it's influenced, but we now know certain specifics, which is kind of nice. Because, for example, lipopolysaccharide is a very bad guy. That is when you have organisms in your gut that carry that, we consider those guys pathogens. The little molecules that bacteria have on them that they release into your bloodstream, some are hormones, right? They're bacterial hormones that influence you positively. You've seen that through the 4 pathways. But pathogens do the same thing using bad compounds like lipopolysaccharide and other MAMPs, and it's microbial-associated molecular protocol or process. It's a weird name that they started naming it, but things that are released by bacteria, and they can be small molecules, they can be fatty acids, they're released into your bloodstream and they create havoc to one of those systems. They alert the immune system, they alert the nervous system, enteric, and so on. So they can be good and bad. That's just a quick, the HPA axis is central, and of course there's neurotransmitter signaling. I want to go through now evidence. I've referred to quite a few studies and told you they exist. Now we're going to go through some. I'm going to try to go through them a little bit quickly because you can read them, and of course these are, I'm citing citations just to build the momentum that this is no longer theoretical, hypothetical. That's all I'm doing. But we want to look at all the variables that belong in this realm now that we've finished the Human Microbiome Project, which is HMP. We've got the MGB, HPA axis now, and that can be even expanded into further endocrine. We know diet is a key factor, key factor. That's your prebiota. We know genetics is a factor. There's actually genetics. And when we meet again at our next seminars, we'll get more into the genetics because male and female compatibility and other endocrine issues, that's genetic driven with the microbiome. There's a compatibility that we're discovering for some of us, especially as you find your mates and your significant others. There's some genetic as well as microbiome. We won't go into much of that today. And then culture, that is, the social culture is huge because remember, you're being exposed to these organisms. Like right now, if I sneeze, you guys will get some of the stuff that's in my oral microbiome, right? If I've brushed up against you, if you guys have hugged each other or shaken hands, you're sharing microbiome like crazy. And just on your hand alone, you got millions of organisms. So the culture is when you have a healthy society, you're passing along healthy microbiome. When the society is not healthy, you can pass on pathogens quite readily. You've all experienced that. That's nothing new to you all. Probiotics and prebiotics, we know that they now scientifically influence. Let's head into it before I run out of time. Because we'll revisit this, you guys. We got to finish out this after lunch. That's the fun stuff. That's the amazing stuff. That's the stuff you're going to take home and use. This, I'm just filling your brain full of information. And you can't purge it, by the way. I know you want to, but it's really good stuff. So for example, microbiota actually makes neurotransmitters. It also can communicate with your endocrine system, the gut and so on, and the brain to produce its own neurotransmitters. It can send signals for you to produce some as well as it produces some. There are specific species that do that, and I'll get to those in a second. And it can also influence receptors. How many of you guys understand receptors? What are receptors? I need some feedback here. Thank you. Did you guys hear that? They are sites on the cell membrane. So your cell membrane, every cell, and every cell is different, by the way. Your immune system versus your liver versus your brain, they have a set of receptors that are ready and waiting for a message to come to them. So if you have a pancreas cell, say a beta cell, what kind of messages does it want to receive? What's one of the things? Insulin, let's say, right? Or glucagon and so on. These are receptors. They bind a chemical messenger and they do something about it. They make a protein. They change. They go fight a fight or whatever. Those are the receptors are what communicates what's going on in your environment to the cell, to the nucleus, to the mitochondria, and so on. This is still a cellular world, mind you. Even though we're talking about bugs, we are still a cellular healing company, right? You know that. We've all taught on this subject. Well, the cell now is responding. So the cellular nature of your cells is responding to the microbiota through their receptors. The microbiota can affect your GABA A and GABA B receptors. It can tell your body to produce more, which makes it more sensitive, or less, which makes it less sensitive. Would that affect you? What does GABA do? It's a calming neuropeptide, right? So it can affect how you relate to a calming event by the receptors, not just the neurotransmitter. Furthermore, the pathway in the microbiota ENS, I won't go into that. You've heard that. There's a lot of vagal activation. The vagal system is almost brilliantly designed just so that your bugs can communicate with your brain. Parkinson's disease. What's one of the treatments? I read yesterday, because I knew this was a treatment, but I read yesterday, Europeans 15,000 Europeans have undergone this procedure for Parkinson's or early Parkinson's. Guess what it is? They cut the vagal nerve. They cut it. You can live without it, but guess what? They're in for a long, long haul because the vagal nerve isn't. Scientists used to think that it was just this random bundle of nerves that you can live without. And I shouldn't pick on. I'm a scientist, but I'm not. It was underappreciated until now. It had been sacrificed 15,000 times in Europe. I didn't see the statistic in the United States. That's ridiculous, is it not? When what's happening is the microbiome communicating with the brain via the vagal nerve, that's been disrupted for a number of reasons. And they're getting to the bottom of that. They're looking at animals and improving the outcome of Parkinson animals. That's going to find its way into humans very quickly. And again, you guys have the tools. This is not out of the realm of our world. Amazing, amazing, amazing stuff. Now, let's talk about some of the bugs, because I'm going to get to that after lunch. Lactobacillus bifidobacterium produce GABA. They actually produce GABA. Escherichia, Bacillus, and Saccharomyces produce norepinephrine. It's pretty cool, right? You guys know what norepinephrine is? Adrenaline. Candida, Streptococcus, Escherichia. Now, by the way, this Escherichia and stuff, that doesn't mean it, or Escherichia, that's not dangerous, just so you know. Candida and so on, they can produce serotonin. Bacillus can produce dopamine. Lactobacillus can produce acetylcholine. Look at all. Now, that's just the tip of the iceberg. There's a number of organisms that can produce the same thing, and they live in various parts of your gut, which gives them differential access to the brain. When there's certain populations of these, like in the colon, the ascending colon, that have a lot of the enteric nervous system, they can send those neurotransmitters to the brain. And there's now evidence that they make it through the blood-brain barrier because the blood-brain barrier is controlled by hormonal conditions, right? It's opened up for things like microglial cells. And if you communicate with a microglial cell, it can go to the brain, so the microbiota says, hey, I want to produce more GABA, I'm sending it through the vagal. The microglial cell in the brain gets the message and initiates in the brain the production of BDNF. You guys know what that is? Brain-derived neurotrophic factor. That is responsible for cognitive, plasticity, healthy hypothalamus, amygdala. I mean, that is one of the compounds that makes your brain amazing and dynamic and so on. Well, it's under the control of the microbiome to some degree. All right, enough said there. I'm out of time, so let me just wrap up a couple of more things so we can come back and cruise on out. What we're going to do when we come back, just so you know, because I might cruise a little bit fast to get to the meat of this, is I'm going to go through more publications showing you the benefits of some of these organisms and how the studies have moved from the human microbiome to animals to human, and they're clean and they're reproducible and they're just flat out amazing. And we love that that's moving so nicely in this direction. So for example, in diabetic rats, probiotics L. helveticus and B. longum considerably improved spatial memory and the synaptic transmission. So they were measuring synaptic transmission and hippocampal long-term potentiation. So we're actually measuring the electric momentum and the electric movement in the brains and showing how those organisms improved it. You remove those organisms, it declined. You put them back, it increased. How cool is that? Right? And that's just, again, that's just a few organisms. In mice here, these are mostly animal right now, B. longum was vagally mediated. So they found when they measured the compounds that B. longum was introducing into our body were being transferred to the brain by the vagal nerve, which is pretty cool. And the effect was lost when you severed the nerve. When you severed the vagal nerve, all the effect of B. longum was lost. Finally, we got Lactobacillus reuteri enhances the excitability of colonic neurons. And again, they can measure those by inhibiting calcium-dependent potassium channel openings. You guys know what those are? Those are synapses that we've been excited about for decades. We've always thought we've understood them, right? Science actually stopped. Those types of calcium-dependent channels and stuff really wasn't growing much until the microbiome came into play. Because they didn't know what else to go. They're like, well, it's a synapse. Calcium moves across, you get an impulse. There you go. No, that wasn't it. This story is now re-emerging. Last three. Luminal administration of live lactobacillus species moderates in mouse jejunal motility. So it by having those there, the motility, the muscles reinvigorated. When you remove them, you lose motility. We don't like that at all. That blocks you up. The anxiolytic effect of Bifidobacterium longum involves the vagal nerve again. And they had to sever it to prove that. And then finally, so these are references I want to start building before we get to probiotics. These are, and how we can take advantage of these studies. I'm just demonstrating to you that the science, the plethora of science that's been coming out is really allowing us to just jump in full force and just go for this. We don't need to wait. And there's nothing but safety behind it all, by the way. I mean, these are bugs that your body loves, they're part of us, and so on, right? There's no downside. Allotypes, the genus and phylum of Bacteroidetes, was overrepresentative in depressed patients. This genus, by the way, the allotypes, is also associated with inflammation overexpressed in chronic fatigue syndrome and IBS. So there's an example, instead of the good guys, some of the good guys, there's an example of when you get an overabundance of that organism, that genus, your statistical opportunity of getting these issues goes way up. And how do we know that? We study people with these diseases and compare it with healthy and do a lot of statistical overlapping. It's very complicated, it's cool, but it's very time-consuming and complicated. And there's other organisms that do this, right? Not just allotypes. I mentioned some other ones. Some Clostridium species, Enterobacteriaceae, are associated with this. So the story I'm trying to present to you now it's a broad stroke. We'll get into some detail after lunch. And more excitingly, we'll get into what you will have in your possession after this meeting, and then every meeting subsequent until we meet that big dynamic protocol, right, with all the prebiotics and all the probiotics. That is the future of this series and the future of probiotics. And that'll evolve, by the way. Just before I go, this didn't start today. For those of you, there's another product that kick-started the microbiome with us. What was it? Skin colonizer. It kick-started because no other product is living for topical use. Furthermore, there's 3 organisms in that product you can't get anywhere else. Skin-based organisms, right? They live on your skin, they're healthy, they combat the bad guys. When they're there, bad guys are absent. So that's really where this started, and we're going to continue that same trajectory for the next future. I'm excited about it. I'll return after lunch. So you don't eat too much while you fall asleep.